In this presentation, I'd like to introduce the StrathSPACE modeling approach and how we're using that to model mesopelagic fish. There's a modeling challenge involved here because mesopelagic fish, as with many species, are physiologically structured, so they have development that goes from eggs to adults involving large changes in relative size, and they're distributed over large areas. So we need a physiologically structured model and a spatially structured model that represents the full demography of the species. And the spatial scale is very large. It's ocean basin scale, in which we need to represent advection, the movement of the population due to ocean currents, plus diffusion or biodiffusion, the random movements of individuals. Additionally, because we don't know very much about the mesopelagic fish, and we don't know much about the parameter values that would represent them well, we need to be able to run many instantiations of a model in order to test our hypothesis and explore different parameter values and do management scenarios as well. So in order to achieve that, we need a model with exceptional computational efficiency. So our solution is to focus firstly on single species. So we're dealing here with a model that represents one species at a time. And we'll use a computationally efficient discrete space and discrete time approach. In other words, the space in which the population is represented is divided into a grid of horizontal cells. And the processes of movement in biology are updated in these cells at discrete time intervals. Vertically, there are two layers. So we have the surface layer, which is the top 100 meters, and a deep layer about 400 meters for adults. So the surface layer is for transport of eggs and larvae in the young stages of the population. But for the mesopelagic, the adults are much deeper. So although we don't represent the movement in full 3 dimensions, we do represent this developmental distinction between the eggs and larvae and adults. Further, we separate the computation of the physical processes, that's to say movement between the grid cells, and the biological processes, the growth, increase in length of individuals as they grow. To get the transport component, we calculate the probability of moving between cells using Lagrangian particle tracking in the context of a general circulation model. In other words, we set off particles in each cell for each time step, track their movements driven by the physics, and calculate the proportion of these tracks that end up in nearby cells and keep tabs of that, which yields the probability of moving between cells in each time step. The advantage of doing this is that we don't need to model movement as we run the population model. All this heavy computation can be done in advance because the physics drives the biology, but not vice versa. And the result of that is that gives us a very large increase in speed compared to doing a Lagrangian ensemble, like an individual-based model where each individual is represented and is moved around the physical domain, the spatial domain. So how do we represent the physical transport? So we update at regularly spaced intervals, TI. So if C is the class abundance and we indicate C minus as the class abundance just before the update. We have C with the subscript Q, which is a length class, and X, which is a physical location, a grid cell, and TI, the current time. Then we multiply the abundance, the class abundance just before the update by a transfer matrix, which moves individuals from location Y to location X at that time interval. And that matrix, as I indicated earlier, is determined by particle tracking in the flow fields from the general circulation model, with plus a random diffusive component. For updating the biological part of the model, then at each spatial cell and at each length class, we calculate the increase in body length that occurs in a time step according to the von Bertalanffy growth equation. So that's a very widely used and famous model for fish growth, which involves L infinity, which is the asymptotic length to which individuals will grow. L is length, delta L is the increment in length over a time step delta t, and the parameter gamma gives the growth rate. Ordinarily, L infinity is something that's a single parameter and a constant, and likewise with the growth rate gamma. But in principle, if we have information to guide this, we can make L infinity a function of the food availability with some half saturation FH, where F is the food abundance, and the growth rate would be temperature dependent with a Q10 value for the change in the growth rate with temperature. So for each source length class, we move surviving individuals after applying a mortality rate to a destination length class, and hence the population at any given spatial cell grows. If we're in a spawning period, then we can calculate the eggs produced by the spawning stock biomass. So that's the individuals above maturity length, and add these eggs in as a boundary condition to close the life cycle. So we combine both of those processes, that's to say the movement, physical part, and the biological part, the growth, by taking each spatial cell in turn, growing, i.e. increasing in length the population over all lengths over the time step. We remove individuals that die by applying a mortality rate over that time step. And if this growth time is greater than the next transport time step, then we stop, apply the transport update as described earlier, output state variables, and then we repeat. The upshot of this is that it produces model realizations that are in strong agreement with partial differential equation representations of the same process, or Lagrangian ensemble solutions, but much, much faster. So an implementation here, so this StrathSPACE approach for the mesopelagic species Benthosema glaciale. So in summary, what we've discussed before, we have a discrete time and space closed life cycle population for the species. It's a length structured model, so we have length structured population. Growth is von Bertalanffy. The larvae are planktonic. That's to say they're drifting with the ocean currents and physical diffusion, and the adults are biodiffusive, so that's with they're moving at random, but not necessarily driven by physical diffusion. Temperature dependent growth and mortality, and the transport in this case is taken from the NEMO model from the National Oceanography Centre at Southampton for the run from 1980 to 2099, so just over 100 years, with the RCP 8.5 scenario for the forward run for the whole North Atlantic. And the implementation is very fast, and it runs at about 1 to 5 seconds per model year. So outputs from the model take various forms. So first of all, we can look at spatial distributions of the stock. So this is the animation here shows the adult distribution in tons per square kilometre for a run from 1988 to 2050. And as you can see, these are the annual snapshots of the adult distribution as we go through the years and now into the future. Interestingly, there's a lot of spatial variability, and the distribution shifts, is patchy and shifts markedly from year to year. And now we've got to 1950 and back to 1988. The model's length structured, so we can also look at the length distribution of the stock. So this animation has length in the x-axis and the frequency or abundance, population in millions, on the y-axis for a representative year. So it shows the seasonality in the length distribution. So when spawning comes in spring, we see a big peak in abundance of the juveniles, and the total population for small fish increases. I should say that the black line is the length distribution of the total population. The blue line is the length distribution of juveniles, and the green line is the length distribution of adults. So the black line is the sum of the other two curves. So as spawning occurs, we see this peak in smaller fish generated by recruitment, which then move rightwards in the length distribution. We can also look at the abundance time series. So in the sequence of plots that we see here, the top left panel shows the abundance of eggs. Of course, that's against time. So that shows strong seasonality. So the egg abundance for parts of the year is 0. So we just get these sharp peaks. Similarly with the larvae, which is the top right panel in the red. For juveniles, we see similar trends, but slightly less variability. We've always got juveniles at some point in the year, although there's still strong seasonality, and much less. That's the bottom left panel. In the bottom right panel, the green line shows the biomass in tons of adults. In this case, of course, that's much more stable through the year, although there's a little bit of residual seasonality there. Interestingly, the warming that occurs in the forward run generates a decline in the stock. The model is currently being used to generate yield curves for the species Benthosema glaciale and Maurolicus mulleri for different management and climate change scenarios.